All right, this presentation is to give you some background information on Lab 5, specifically exercise D. You're going to be doing exercises A through C as well, but they do not need to go in the paper. Only exercise D, beginning on page 77, is going to go in your paper. I want to give you some background information on the overall goal of this part of the lab so that you can understand what experiment you're doing. So your goal is to use amylase, which is an enzyme made out of protein, to break down starch, which is a carbohydrate. Amylase, I have the structure of it over here, is a protein. Proteins have all of alpha helices, which is what these kind of swirly things are, and some of them have beta sheets, like these lines going in different directions, and they have a three-dimensional structure that is dependent on what temperature it is. You are going to be looking at a variety of different temperatures for this amylase to work at. 0 degrees Celsius is the point where water freezes. 20 Celsius is about room temperature. 37 degrees Celsius is the temperature of your body. 70 degrees Celsius is getting pretty warm. And then 100 degrees Celsius is where water boils. I want you to think through with this structure and all of these different three-dimensional shapes that need to stick together. If you start heating things up, there's going to be more movement, and eventually some of the hydrogen bonds that are holding this structure together are going to start breaking, and your protein could start denaturing at high temperatures. And at low temperatures, things are moving more slowly. And so what effect do you think that is going to have on a chemical reaction? Now, the starch you are looking at, we are using the IKI detection, which detects starch, which is the starting point. So we have starch, it's going to break down with our amylase into, I'll finish writing starch, into smaller parts. And so over time, starch content, if the enzyme is working, is going to decrease because there is less of it around. And so because we're detecting starch as the experiment goes on, you're going to be getting lower numbers because you have less starch around. Now, I would like you to read very carefully pages 77, 78, 79, and then at the top of page 80, it has you write a hypothesis. I want you to think very carefully about what you are testing and write a clear statement about what you think is going to happen. Now, that statement could be what temperature you think is going to work the best, could be what temperature you think is going to work the worst. It kind of depends on, there are a variety of hypotheses that you could have for this. I wanted to give you an example. So if you read through parts A through C, we are detecting protein, monosaccharides, and starch. And a hypothesis, so if I bought some milk and want to test it, I would hypothesize that milk will test positive for protein, positive for monosaccharides, and negative for starch. And that is based on my knowledge of what milk is and what it contains. I could write up some background on, what milk is and how it contains nutrients for young cows or people to drink, and why my hypothesis is that it would contain protein and monosaccharides, and why it wouldn't contain starch. So your hypothesis, you want to think about writing a clear statement about what you think will happen. Then you're working through testing your hypothesis, writing up some experimental designs on pages 80 to 81. Now, the experiment you're doing is written out nicely on pages 82 to 83. So you might want to read pages 82 to 83 before you write out your experimental design. And basically what you're doing is you're going to be incubating amylase and starch together at various temperatures, and at different time points take out a sample and use a spectrophotometer, which is going to absorb light at a certain wavelength. The test will be dark when there's lots of starch around, so your absorbance will be high, and then over time you're going to lose absorbance. You could also do this in percent transmittance. If you do that, then it's measuring how much light goes through, and so then your number is going to be low at the beginning and high at the end. Make sure that you know if you're writing down absorbance or percent transmittance, because they read opposite things. Then pages 84 to 85, you're going to analyze your results, think through if you should accept or reject your hypothesis, and then write a conclusion. You are going to write a paper based only on part D of this experiment. I highly recommend that you read and fill out pages 77 to 85 carefully, because that's going to help you write your paper. The order that things go in are on pages 87 to 88. You're going to start with a cover page. Please come up with a title for your paper. The title should not be Lab 5. Come up with a descriptive title for your paper. Put your name and your lab partner's names on the paper. Your name goes first, then your lab partner's. And then also put the due date and the date you submitted it, which might be the due date, that's fine. Then you want to start with an introduction. I like to draw the kind of two trials thing. Your introduction is going to start broad, so maybe talk a little bit about what an enzyme is, what amylase is, what starch is, and you're going to narrow down to your hypothesis. Now at this point you should have already written your hypothesis, so that's pretty straightforward. That goes at the end of the introduction. Then you have a materials and methods section. This can be kind of a list, a stepwise list is fine of what materials you used and how you did the experiment. You can refer back to pages 82 and 83 to describe how you did this experiment. Then you have your results section. Your results section is going to include a data table with your numbers on it and a graph. You can hand draw your graph, that's fine. Or if you know how to make a graph in something like Excel or Sheets. Now you'll notice I drew one over here. Should have time on your x-axis, and then activity or absorbance or percent transmittance y-axis, and indicate what's happening. Your graph will have more than one set of data because you are doing more than one temperature. And then you want a figure legend, which is this figure 1 enzyme activity over time that I wrote down. Your figure legend is going to give a number for your figure and then a short description of what the figure is. Your tables should also have table legends, say something like Table 1, amylase data at various temperatures, something like that. So that's going to be your results, and then maybe there's a sentence or two, actually there should be a sentence or two about what the results are. So results in figure 1 and table 1 show that amylase activity worked best at blah temperature. Then starting to broaden back out, as you can see from my triangles, you're going to discuss what do the results show and maybe connect that back to your introduction. So the results show that amylase worked better at this temperature. That makes sense because amylase is a protein, and connect it back to what you talked about in your introduction. You can also talk about if your hypothesis was accepted or rejected here in the discussion. Did your data support your hypothesis, or do you need to reject your hypothesis based on your data? Then you're going to kind of zoom out even more to your conclusion section, which is going to be just 1 or 2 sentences. You know, our conclusion from this experiment is this, and again, maybe if you accepted or rejected your hypothesis. Finally, you need some citations. You should cite your sources. Your lab manual should be one of your sources because that's where you're getting all of this information from. Your textbook might be another good one. You can use the internet to find other sources. Make sure they are reliable websites though. So Wikipedia is not a good one. Look at what website you're doing. Is it a real scientific website, or is it someone's blog? Someone's blog is not a good resource. So make sure it's a reliable website. And just so you know, you're going to be turning in this paper on Blackboard, and I have anti-plagiarism software that it's going to run through. So if you've decided that you're going to talk your lab partner into sharing their paper and you're going to submit it as your own, then you are both going to be called in to talk to me, and we're going to come to get to the bottom of what happened, or you're both going to get zeros. So please write your own paper. I don't like dealing with plagiarism, but I will if I have to. The paper is due on October 31 by the end of the day, so it'll be 11:59 p.m. You're going to turn it in on Blackboard. There will be a link for you to turn in the paper. You're not turning in a hard copy. If you have questions as you are writing the paper, please send me an email. I'm happy to answer questions. Your paper, a well-written paper like this, should be 2, maybe 3 pages. It does not need to be, that's not including the cover page, so the cover page should be separate of text and figures. You're looking at 2 to 3 pages. It does not need to be a 12-page novel of this. 2 to 3 should be sufficient for what you're looking at today. Thanks.